Hello?
Hello, hi sir.
Hi, how are you?
Fine sir.
Good. How are you, Dick?
Oh, sleepy. Otherwise, okay.
Yeah. Didn't know if we, I had a little technical difficulty getting into the Hangout, so.
Oh,
Yeah.
okay. Okay, fine.
Yeah.
Well, anytime you're ready.
All right. Any announcements anyone wants to make before we start?
Nope.
Okay. I don't know if we'll have other people showing up, but, oh, there's Mayukh. Okay. All right, welcome to the meeting. So today we're going to have updates on GSoC, and we'll have maybe some papers, and maybe we'll have a presentation after that. So why don't we start with who wants to start, Ujjwal or Mayukh, on their weekly progress?
I can
Okay.
share this.
Yeah, you can start.
Let me share my screen.
Okay.
My screen visible?
Yes.
Yeah. So basically, I have made from the. I guess we can surely close some issues that are being on the board today, and I have completed almost all the work on the front end, and I have pushed it to the repo, and I have taken some of the my my classmates and some professors have given me some suggestions on what should I do next on this. So these are some points from there. Like most of the people like the architecture and the JS or JavaScript that we have embedded in our portal, and like I have got a suggestion like Splash Page 2 is hard to read because of the contrast of colors. So I will get today and make it better. It will not take so much time. So this is the one of the major suggestion. So to do like this week after Wednesday, I guess I will start working on segmentation models. I have started incorporating all the front end with the Django framework. Like there are lots of static file and HTML files, so it will be completed by Wednesday for sure.
Okay.
These are things that have been done till now, and these are the things that are on agenda for the coming weeks. Like for till Wednesday, I'm going to complete all the Django work and start working on segmentation. There are some minor glitches on dashboard which will be sorted out by tonight, I guess. And when I have started working on DevoZoo, so I have some question regarding DevoZoo. So like I have seen the DevoZoo data. Some of the data is in the format of Excel files. So should we just we can also make it display completely on our website. So it will be displayed like the whole data in form of tables on the website itself. So a person can come view our data, and if he wants to download, he can download. And
Yeah.
my second question regarding Zoo is, should we like try to keep a track like who is accessing data? We can do it like we can ask for the email addresses and send the download link in the email itself. So these are the two questions regarding it.
The first one, yeah, we should make like multiple versions of the data. So like, I'm not sure which ones you're looking at. A lot of them are in CSV format. I guess some of them are in Excel format.
Yeah, most of CSV.
Okay. I don't know what.
I'm here.
Yeah.
Yeah, I think I have an opinion on this.
Okay.
Like, can you hear me?
Yeah. Yeah.
Ujjwal?
Yes, yes, yes.
Am I audible?
Yes.
Yeah. So what you can do is like you can make something like Kaggle where they show just the first 5 or 10 rows of the data so that they get an idea.
Yeah,
They
that's
see the
something
first
I
few
told you.
rows.
Right.
And to download the whole thing, and to download the, like, they will get a small snapshot of what's in there.
Mhm.
Yeah.
Yes, that's actually like I'm thinking to give the idea
And
of
then,
website or the idea
yeah,
of the data.
if they want, they can download.
Yeah.
Yeah.
If it's possible. I don't know much about web, but still. So that the users will get an idea before downloading. And I think we should move everything to CSV if possible.
Yeah.
Instead of, like, there are some in Excel files, some are in CSV. We should move it to one or the other format. And I think CSV is much more like universal than Excel.
Comfortable and compatible, yes, I agree.
Yeah,
exactly.
that's pretty universal. I don't know, like,
Yeah.
I don't know what other formats we might want. I don't know how hard it is for Ujjwal to convert things across different formats. I know we had done
it
some
won't
stuff.
be very hard.
Very hard?
I volunteer for it. If it's required, I can volunteer for that stuff.
Oh.
No, it won't take much time.
Yeah. I don't know.
I can volunteer
we can talk more about it, like, for
for
another
that if
time.
required.
Yeah. But, I mean, I think for now,
So, can you just add a little bit of on how the user can just run local machine?
Yeah, sure. I'm going to add it all. Like
I
right
kind
now,
of had for that file.
yeah, like right now it is like
Yeah, because it took
functional.
me some time
It is
to
not
know
functional right now.
where's that main HTML files. So just
Yeah, exactly. So like when I will connect all these files to the framework, then I will upload all the documentation regarding it.
no, just add some basic documentation that run this file to check, like, to run it on a local machine and see how it goes.
Yeah,
Because I was
exactly. I'm going to.
trying to run it on my local machine and it took me quite some time to find for the, like, real file.
Yeah. So like when like right now it is not complete. Once it is completed, I will like add all the document.
I get it.
Sure.
alright.
Yeah, fine.
Yeah. And so, I mean, I think the format for now is good. We can, talk about maybe making, like, a preview for data. So, I think we can focus on, like,
I think there are two of that.
What was that?
I don't know much about web, but as far as I know, like, there are plugins, I guess.
Yeah, I mean,
And how is the data hosted? All of the data is hosted on Drive, Google Drive, right?
a lot of it is. Some of it's hosted. There's some on GitHub actually, like as CSV files. So I mean, we might revisit like where things are hosted. Now some of it is video files, so those are I think the ones on
So
Google Drive.
I'm thinking like for the data which is hosted on GitHub, we can just give the raw link so that it can be opened in the web browser itself, and they want to download, they can download it. And for the data which is in Drive, we can surely give the download link or like view link or something like whichever suits which kind of data. Like for video, we can give view link so that they can download it if they want. And for the text data, we can give simple download link. So
Yeah,
my
it's
main question is around like from what I'm getting from the discussion is like I'm going to show a quite snapshot of what the data looks like. So if the person wants to download it, he can
yeah,
download it.
it's like Kaggle. Like, I
I
think
get
Ujjwal
it.
has seen those things. Ujjwal has seen.
The second thing is like should we keep track who is accessing our data or not?
If it's not too hard to implement, that would be a good feature. I don't know how much use it would be though. If it's going to be like really involved.
Okay, sure. I can simply add like we can enter their email address and I can
Maybe
automatically email
yeah.
automatically email with the download link and it will be added in our database as well.
Yeah.
The next topic is like DevoWorm Academy. So this is something I need to be updated about. I don't know what is happening in that context.
Okay. So that's something that so that's something that we have kind of on GitHub. So it lives on GitHub, but it's not. I haven't really thought out like how to host it through this set of websites. So I guess we have
Like we can easily provide like DevoWorm Academy or tutorials, study materials, or if we have some video demos through the website, if you want,
we could do
and we can like
we could start with just like links. I don't think it
yeah.
needs to be integrated with this website too much. Although, we could just have like maybe an introductory page, kind of recapping all the resources. So we have the DevoWorm OpenWorm curriculum, which has a number of lessons in it. Then we have the DevoWorm ML, which is the series on ML, and so I think those two would be, and those are both on the GitHub in the GitHub repo under those names respectively. So maybe just link to those things and maybe give a little description about what they are. We can go over the copy on that when that comes up. Just when you submit a pull request, you can bring my attention to it. I can look it over.
Oh yes, yes, yes. I will surely do that. So, and the last task in my agenda is Blender work. I'm doing it like every Sunday for like 2 and 2.5 hours. So I will do it this week as well.
Yeah.
So these are the changes like what I have got, and from the discussion I have got some few points which I have doubts about. So I will readily implement it. Also, I have hosted my blog. Like I have written all my blogs. Like as the sir has said, there is some spelling mistakes in the blog because I am writing directly in the Sublime text. So I am writing first in Grammarly and then I will host my blog. So I think I have written two, three blogs. Uh, yeah, two blogs are hosted, and rest of the blogs will be hosted by tonight. So this is the update on blogs, and this is the open one. Like it is not completed yet, but this is something like everyone has seen, and I would like to get like if there is some opinion or something they want to change. So I will quickly go over here. So this is the main page. Then there comes the C.Elegant models. Then there is digital bacillary model. Then there is gender segmentation tool. This is something that I have incorporated in the project from a long time, and now I think Krishna has also stated his interest in this work, so we can make a good model for it now. Then yeah, so DevoWorm, DevoZoo methods. This is something I am creating, and I am creating the different pages for DevoZoo and Devo method as well. And DevoWorm Academy, we will see what we have to do. This is the federal history. Then these are the numbers. There is the founder page and team members niche which needs to be updated.
Yeah.
And this is like a contact space. So pretty much the website is same as shown earlier. So once it is completed, I will go through it all, and I guess these are the updates from my side.
Okay.
If anyone wants to add something.
We had, I guess, Jesse, I think, asked me about how do we submit suggestions for the website work. So let me actually show this how for now we might do this. So let me share my screen as well. Thank you. I'm just gonna go to this thing real quick. So this is the I think it's under GSoC 2020 in that repository. If we can get in. There we go. So there's a projects. If you go to the project board for GSoC, And this is, yeah, so if you want to make a comment on like a design evaluation for the layout for Ujjwal's work, you can put in a comment in this board. Or
the screen isn't being shared right now.
Oh, it isn't shown? Alright.
You're not presenting.
Yeah, it didn't. I thought I did, but it didn't work. Alright, you can see it now? Alright. So we're in GSoC 2020, we're in the project board. Basically, you know, if you want to share some feedback, you maybe want to become a collaborator on this board, so I can invite you. And if you are, then you can put in a comment here. This is a design evaluation for Ujjwal's layout. This is a generic comment, and it has this label Open DevoCell. So if you look in GitHub, the labels are here. We're putting this label, which I made, on those issues, and then we can go through them systematically in future meetings. So if you have an idea, I mean, you can send me the idea for like an update, and I can put it up as an issue, or you can do it, and then we'll have them all under that tab. So Ujjwal can actually look at for those. He can search for those labels and see what the feedback looks like. In terms of feedback, I mean, the only thing I'm still thinking about is like the visual elements issue, which is like where we have all those splash pages in the background, and they're like different colors, and I don't know like if that's really distracting or not to people, or if it's a little incoherent visually. I mean, I think maybe a solution is to make it like a uniform sort of mask of a certain color, so like a very transparent light green or something might be good.
Oh, okay, so like, I get it. Like the image is really flashy for it right now, I guess.
Well, yeah.
So I will try to make it more realistic.
Well, it's just that like you go from like a really dark blue image to like a colorful image to like a green image, and then it's like, you know, it's distracting sometimes when it goes like, you know, there's that transition. But if you have like a green, have you ever done a transparent mask over an image?
Yes, and I got it. Like you are saying, like whatever the image in the background is, you have to just put a mask over it, like some sort of green transparent mask, so that it will not go two different in colors.
Yeah, so it's just like kind of like it's part of the template of the site, like it's part of the color scheme.
Yes, sir.
But it isn't like this. It isn't jarring, you know. I mean, otherwise, I think it's good to have like different backgrounds coming up with different topics, and I like the way it's laid out. With
I will do that.
Yeah. So, and then we'll talk about the copy, like, you know, for like the academy, and then we can talk about how to set up the data. Like, I think for the data, it's just a matter of linking to the data sources. Maybe specifying that they're mostly CSV files. I think that's good to just have it in CSV format because I think it's pretty universal. You can go from that to a lot of other things.
Okay.
Yeah. Thank you, Ujjwal. Mayukh, do you want to do a short?
I'll check my screen.
Okay.
Yeah, can you see it?
Yeah, it's coming up.
Yeah, so since the last week, like the initial part of the last week, I actually did some PCA, it's like principal component analysis, and we talked about it, right?
Right.
We talked about it last week. Yeah, so this week I started out with the segmentation model. So I went with this data set. This is the warm image database. This is out there online and it's openly available. So like all of these are pictures of the C. elegans embryo, and in the middle those little white patches are they're like the nuclei of the cells. So I tried to build a model that could segment out the nuclei from it. So the problem was that there was no like data. So what I did was that I had to first initially I just laid out the pipeline, and what I did was suppose like we take this picture of like one of the sample pictures, then I just drew circles manually over it, and then I augmented it really like in a lot of ways and generated quite a bunch of images. But then I scaled up this process further and I drew circles manually on like 70 original images, and then I got a lot more images by augmenting. Yeah, so after that, after training all of that, I kind of went on to train a model which is based on the ResNet 18 architecture, and like I'll just keep it short. Yeah, so then like I won't go into these details. Like so then like you see this plot?
Yeah.
Yeah. So the left, the row to the column to the left are the original images. The column to the middle are the predictions made by the model, and the column to the right are the original like labels. And this is like trained on the augmented data set. So like even though it's not trained on all the images, it's trained on a very small slice of the images, but it's still performing pretty well on the test set for now. So I want to improve on this over the next weeks. For like so that would be I'll be trying like for now ResNet 18 is like my dummy architecture. I'll be playing around with bigger and smaller architectures and see which would fit the best. Yeah, so that's pretty much it.
So in this set of images here that you have in the notebook, so the ones on the right are what are the ones on the right?
The ones on the right are the original like the original mask, the actual positions of the nuclei which were labeled manually by me.
Oh, okay. So you're doing manual labeling.
Like I did manual labeling for only 70 images, and then after augmenting I got a lot more.
Oh, okay. All right.
Yeah, so these images are already like transformed and augmented, and like we discussed about it in the proposal.
Yeah.
Yeah, so like I think you can see I turned it backward, so you can see the labels like original mask, prediction, and input image.
Yeah. Yeah, we talked about this on Slack. Yeah. So this was like hand labeling and then masking. So like last year we did the masking, and we've also done hand labeling in the past. I think that's probably the best way to get a good, at least some approximation of what the cells should look like to the is training.
Just looking at the images right now, so like the results are great, but they're not like 98% great. They're like I don't know. They're like 92%.
Yeah.
So I'll have to improve it. But it can just improve by getting more data. That's it. For now, I'm just prototyping on a small amount of data so that I can just test things out faster.
Right.
Yeah, that's pretty much it. Like you can see the predictions in the middle. They're like pretty, like they're convincing.
Yeah.
But they could be better. Let's see.
Yeah, there's one where the two cells are merged, but that's just kind of because they're close together.
Yeah, so like it merged a little. I don't know why. It's because like they're heavily augmented. The images are also heavily augmented. So yeah.
Yeah.
It'll learn more the more it trains. I didn't even like train it for long. I trained it for like 15-20 minutes. So this is like good. I'll try with a lot more things. I'll try with those techniques I talked about in the proposal, and then probably I will get better results.
Yeah.
Yeah, so that's pretty much it.
I was just trying to think of other ways you could do this to improve or boost the performance. I think those are the two best ways. I mean, there might be, with ResNet though, you can't necessarily use adversarial data, which might be a strategy, but I don't know if that might be overkill for what you want to do here, because it's, what's the adversarial form of like a bunch of cells embedded in the background.
I get it. Like you want some kind of a generating model, right?
Well, that's an alternative. I'm just thinking about how you could improve. Yeah.
Yeah, we can actually in the future. I've worked on this kind of like using adversarial networks to like make more data, right?
Yeah.
That's what you're talking about. Right? Yeah, we can work on it. Like for now, augmentation is our domestic solution, but we can work on something like that later on. Like I should first prioritize on getting a good accuracy in this kind of a model.
Right. Yeah, I would agree. And just like try to work, because if you have more data and more training
I want
time.
to make it like, yeah, I just scale out the data now. That's it. I'll just multiply the data. That's it.
Yeah.
I think there's a lot more images. There are around 200 images, so it can like get a lot better. 200 original images. So you can imagine how large the training database, like the training set would be. The training set would be much larger because augmentation.
Right.
And like it's not even, I'm not even storing the augmented images. They're like transformed on the fly. Like every time I show you, wait. Like when it's being augmented, there are certain probabilities of each transformation happening. You can see here, right? For now, I'm using elastic transform p equals to 1, which means like probability is 1. Every image will go through this transform. Vertical flip p equals to 0.5. So only half of the images. And so like if we consider all of these probabilities, then every image is going through a random set of like a random permutation of these transforms, a random combination, a random subset. So every time an image goes through this augmentation pipeline, every time it's like becoming a different image, a unique image. So I'll try and play around and make this augmentation thing, like I'll add more stuff and see if it goes better or worse. I'll tweak with these parameters and see how it goes.
Okay.
Yeah, so that's pretty much.
So like I would like just add one point. The parameter augmentation that you are talking about is pretty nice way to get results, but to get it in a more precise way and in more and more values that humans can do, you can either input these values randomly, like first of all try to determine like what is the upper threshold for these values before it gets completely bogus.
Before we start using information. Yeah, right.
Yeah, and then you can try to add random more and more values. I guess you can use Git plus CV.
That would be my work for the next week, I guess.
Okay.
Sure.
The coming. Yeah. So that's pretty much it. I'll just drop the blog post link for my
Yeah, thank you.
last week.
Oh, this is the blog post? Okay. It's pretty good. All right. Any comments on that? Any questions? Let's see what we have in the chat here. Jesse, I was going to look at some Hello, yeah.
Oh yeah, sorry. I think I was muted the whole time. I was trying to talk something. Yeah, with Mayukh's work, right? I just wanted to ask one thing. Like when you do the data augmentation, are you also taking care of the brightness balance and stuff like that?
For
So
now,
that-
like
Yeah, I actually asked that because hello.
I'm just randomly changing the brightness by a factor of 0.2 for now.
Okay. So
Because like the data, see, I'll just drop the link to the dataset as well. Like even in the dataset, original image dataset itself, the brightness varies. Like some of the images have really low contrast, some of them have really high contrast. So you can have a look yourself. Yeah.
Okay.
Yeah, these have been captured from different sources. So
Yeah, regarding the contrast thing, I think you're aware of this contrast stretching, right?
Yeah.
So probably that can be very helpful. Before inputting those images into the model, like maybe you could try and add the contrast stretching thing in the pipeline and see how the performance is improve if it improves the performance or not. Maybe you could try that. It's just a suggestion from my side, because based on my experience, it worked out really well in some other project that I was working on. Because when you start when you move this to production, and when people actually start to upload their own data sets, so there can be a lot of variety of now you have a plain background, white background of image, right? So those can be inverted. A user can try to input the inverted images as well. it will become a black image, but it would still be the same image. Like the background would be black. So if you're anywhere in your pipeline, if you are applying some kind of thresholding or any kind of noise removal technique, then in those areas, this brightness level plays actually it can mess up things. So I can like it's just a suggestion from my side that if you want to explore.
You're right. I'll have to do that. Yeah, thanks. I'll do that.
All right.
Yeah, that's it from my side. I had that question.
All right. So let's see. We have Jesse had a comment. I was going to look at some DevoWorm Academy, actually, but I'll have more to say on Slack. Limited discussion ability right now. So yeah, please look over the stuff like the DevoWorm Academy. I think is just the DevoWorm ML and the DevoWorm OpenWorm curriculum material. So please do. And Vinay says, "Hey Ujjwal, can you please ping the website link if it's hosted somewhere?" So I think right now Ujjwal has it like in a private. Well, he has it in the GitHub repo for GSoC 2020. So I don't know.
Yeah, it's there.
Once I completed this whole website project, I will host it on Heroku app for sure.
All right.
Oh yeah, great.
And then Jesse says, "Is there anything in the academy specifically about machine learning, DevoWorm ML?" I mean, we just have like the ML, like the ML course materials. I don't know if there's anything more specific than that. I mean, we can create materials, but yeah, I mean, it's a little bit like right now. Yeah, it's a little bit uncoordinated yet as to how that's going to fit together. Maybe if you could give some suggestions of how we might make that more like salient to learners or to people to understand. Maybe make it more relevant to them. Then Mayukh's blog post. Vinay says he's muted. He was, and then the worm image data, which is the source data for this Mayukh's work. So that's, and check out Mayukh's blog post. I'll open that up for later. Yeah, so that's, thank you for the updates, everyone. And that's good progress. I think, especially, well, I mean, Mayukh has like, looks like he's also done some work on the movement aspect. I don't know if he showed that part of it in the meeting, his notebook on movement, cell movement. I think that was on one of the Friday meetings.
It was one of the older meetings. Yeah.
Okay. All right. So in coming weeks, we'll probably revisit that. We'll revisit the website, of course. So next thing I want to talk about is, I guess I'll go to the papers for this week. Let's see if I can share my screen. And that. That's, yeah, there we go, select that. Okay. Can you see my screen coming up? Should be coming up here. All right. Can you see my screen? All right. Good. So the first, we'll go through two papers here today. The first one is, if I can open it up. There we go. So every week I like to share papers on different things relevant to development or machine learning. This one is shared developmental program strongly constrains beak shape diversity in songbirds. and this is by a number of authors. So this is about, so the abstract is the striking diversity of bird beak shapes is an outcome of natural selection, yet the relative importance of the limitations imposed by the process of beak development on generating such variation is unclear. Untangling these factors requires mapping developmental mechanisms over a phylogeny, which is an evolutionary relationship, far exceeding model systems studied thus far. And so, if you're familiar with Darwin's work with in the Galapagos, he looked at beak shapes of finches as one of the examples of evolution and how evolution is mutable and gives you different outcomes based on the ecological niche of the organism. So that in this case, what they're interested in is like looking at just the shape of the beaks and understanding the shape variation. I think last week I actually talked about Darcy Thompson in On Growth and Form, and I talked about those coordinate systems that he would overlay onto different fishes, and like he'd overlay it onto one species of fish, pick out some landmarks, and then take that grid, put it on a different fish, and stretch the grid so that they matched the landmarks of that fish. And then you could have like these warped coordinate systems that would describe the difference between the morphology of the different species. And so I bring that up because in this paper they address the issue of beak shape diversity using this comparative morphometric analysis, which is where they look at the morphology, they measure it with some metrics, and then they analyze the differences. They analyze beak shapes in a diverse group of songbirds. Here we show that the dynamics of the proliferative growth zone, which is a part of the beak in development that controls how the cells sort of form the beak, must follow restrictive rules to explain the observed variation, with beak diversity constrained to a 3-parameter family of shapes, parameterized by length, depth, and degree of shear. So we're talking about the length of the beak, the depth of the beak, and then the degree of shear, which is a mechanical property. We experimentally verify these predictions by analyzing cell proliferation in the developing embryonic beaks of the zebra finch. Comment here. Okay, Jesse, I mentioned the Thompson Forms book to Anson, already some of his development of his ideas. I mentioned he'd come here, but he wasn't able to join. That's okay. You can check us out on YouTube, or we can talk about it in our meeting. But yeah, we were talking about that with Anson, who's in my other group. That was different thing. Okay, so our findings indicate that beak shape variability in many songbirds is strongly constrained by shared properties of the developmental program controlling the growth zone. So let's go down to the figures, because those are often more enlightening than just reading text. So, first of all, they talk about they introduce this, and then they talk about the results. So one of the things they do is they quantify this beak shape diversity. And one of the things they're doing, of course, is they're using a mathematical model to represent the beak shape. And so, but what's important here isn't just to characterize the beak shape, it's to characterize the difference in beak shapes. And so they've been able to figure out the most important beak shape parameters, depth, length, and curvature. And they can study this from 2-dimensional profile images. So they can take pictures of beaks just kind of like out of maybe a book or off of Wikipedia and just estimate these parameters from those images. So then they've been able to do this across the diversity of beaks of different species within a genus Geospeza. So this is a lot of different species. I'm not familiar with that genus, but it gives you a fair amount of diversity. And then they also can use, in their previous study, they used computed tomography scans. And they use the scalings from these to sort of calibrate their estimates for the beak shapes in the 2-dimensional case. So what they're doing here is they're sort of understanding the beak like as a three-dimensional object, and then they're applying this to a bunch of two-dimensional images, because they know, once you know like certain parameters of the beak in a three-dimensional model, you can apply it to a broader class of two-dimensional models. And so then they do these transformations where they have, let's see, they're basically doing a bunch of mathematical transformations to find the difference. They're finding a derivative profile, and then they're actually using like, I guess a morphospace approach to understand the sort of how closely the two shapes are related. So they're mapping these shapes and the parameters to a space that's sort of like, sort of like gradient descent, but it's looking for how closely they're related. So you don't want to just compare them sort of discretely. You want to put it into a coordinate space that says, all these parameters are related is like maybe a three-dimensional space. Now, if we have this as a three-dimensional space, where do our beaks fall out? They fall out in different points that we can then find that's a space that's differentiable. So you have the three-dimensional space, one dimension might be beak shape, and you'll have like a range of values, and then beaks will be distributed along that set of that distribution. In the beak shapes will be within that distribution, and there will be two other dimensions, and then you can just simply make calculations between the two beak shapes. And it doesn't necessarily matter if they're how closely they're related in evolution. It's just that those relationships are purely quantitative based on the data. So this says nothing. This says a little bit about evolution, but it isn't like it isn't a surrogate for evolution. In other words, if there's a change in beak shape, that doesn't say that like evolution, doesn't give you an evolutionary distance necessarily. It just tells you that there was some differential morphogenesis in these two beak shapes. That could be due to developmental pressures. That could be due to evolution, but we don't really know. So this is kind of the idea here. So you get these different beak shapes, these triangles, and they give you these different, basically forms of triangle. And they're characterizing these different clades. So these are groups of species that are evolutionarily related that they can take and they can say this is the beak shape for this group of species. So the phylogenetic and morphometric structure of beak shapes. And so, let's see, this figure is actually, okay. And then they basically talk about the different parameters. See, the hierarchical collapse of beak shapes onto each other under scaling and shear. So they do these pairwise comparisons of beak shapes across phylogeny. So all the beak shapes are compared pairwise. And then they say a heat map of E sub S resulting from pairwise comparisons of all beak shapes in the phylogeny scaling one beak shape onto another. Crosses indicate pairs of species of beaks do not collapse by scaling transformations, as there is no minimum E sub S as a function of the scaling factors. So I think they're looking at the ones that's like the beak shapes that scale together. So, it's like you have this developmental scaling. It's basically a scaling of growth. And so if they belong to the same group of beak shapes that scale together in terms of their growth, then they include them in this analysis or don't include them in this analysis. Let's see, what is this? This is the basic pattern of beak shape diversity. Okay, so this is probably more informative. So you have these different beak shapes, and there's the scaling factor. So in other words, you have this relationship where they scale in a consistent way. They might be different shapes, but they grow in the same way. And then you have that as a group of beaks. And then you look at shear plus scaling. So you have like this growth scaling, and then you have this shear scaling. And then you evaluate them together. And so what they say here is on small phylogenetic scales, beak shapes collapse under scaling alone. For example, the two Geospiza species on the top left, creating groups of similar beak shapes represented by colors. These group shapes in turn collapse onto each other under shear in their length direction. So specifically, all group shapes collapse onto the shape of the blue-colored group. So they all basically have some commonality under this blue group. The blue-colored group can be approximated to an extremely high precision as a section of a parabola, as shown on the right. That's what they show this parabola here. The combination of this hierarchical collapse under scaling and shear onto the blue-colored group and the collapse of the blue-colored group onto a parabola leads to the conclusion that all beak shapes are considered as conic sections. So what they've done is they've taken all the beak shapes and they're explaining it in a number of different ways. They're saying that there's this scaling commonality amongst certain beaks. There's a shear commonality. In other words, they have common, they have related attributes in terms of scaling, in terms of shear, and then this parabola, which explains the rest of the variation. And so those three factors will explain why there's so many different types of beak shapes. If they all come from a developmental precursor, there's a lot of variation, and these are the three reasons why you have these different beak shapes. And so this is an example of experiment testing the time development of the growth zone in developing zebrafinch embryo beaks. And so this is basically a picture of the beak as it's growing out in development. E5 through E9 are the developmental stages in the embryo. And so you see these shapes. The beak will start to extrude from this E5 version, which is this kind of like a bump. And you can see this red circle, and that red circle is this growth zone. So this is where basically a lot of the growth is happening. So you can see in E5, the growth zone is rather large. E7, it's a little smaller. And then E9, it's very small. And this is where a lot of the cells proliferate in this growth zone. So the cells aren't just growing everywhere. They're growing out of this growth zone, and then they're migrating out to the edges, and the shape is being influenced in that way. So the shape is modulated throughout several developmental stages in terms of how it's extruding from that little bump here. And so this means that, given different species, given different variants of genes, given different chemical factors, this can be controlled in different species. So let's see. That's probably enough for that paper. So let's see. Dick said assumes independence of parameters. I think they do assume that. I'm not sure what their controls are for the parameters. But I think, yeah, they do assume that independence. And there are a lot of methods. I don't really know what the beak shape analysis is here, and then, yeah. So I'm not really sure how to answer that question, but I am going to share the folder. I can share the folder here in the chat, and then people want to look at the papers in more detail. There's another paper that I have here. Evo 4 becomes 2. Devo 4 becomes 2. So this one is actually, this is a dispatch from Current Biology. And this is actually quite a different area. So the abstract is wings and halteres are homologous flight appendages. And halteres are in insects. Flight appendages whose shape differences are controlled by the UBX transcription factor. Recent research shows how UBX regulates apical and basal extracellular matrix proteases and their inhibitors to achieve this morphological divergence. So that's a lot of jargon, I guess. Basically, what they're saying is that there are two different structures that they're looking at here for, is in terms of like wings. So, halteres are sort of the developmental precursor of wings. And they talk about this in terms of like how things are regulated at the molecular level. That there's a really sort of fundamental regulation of how wings are shaped, whether they form or not in different species. And so the dorsal thoracic appendages of flies differ, which is where the wings are on flies, differ in size, shape, and cellular morphology. Previous research analyzed how the UBX or ultrabithorax Hox protein controls patterning, cell proliferation, and cell differentiation. And so to regulate the differential development of wing versus haltere. A recent paper in development now completes this study by analyzing how UBX modulation of the apical and basal extracellular matrix affects their morphogenesis during metamorphosis. And so this is one of these papers where they feature a couple of papers that are, highlighted in that issue. So this is an example here. This is a graphical summary of the paper. You have wings and halteres, and so you have this control of UBX on this. You have formation of, or you have this haltere structure, you have this wing structure, and there's a difference in the way they're shaped by this factor. So that's that one. I just wanted to go over that one quickly because I didn't want you to be puzzled by it. So one more thing I wanted to talk about before we go, which is that we got, there was an invitation for a new biosystems special issue on, I think it's on let's see. So we were going to do this paper a long time ago on periodicity in the embryo. And so it was on this idea of differentiation waves, which are waves in and I know Dick is involved in this, at least at some level. I wasn't sure, but I got the invitation on my email. It's about the idea that there are waves in development that contribute to differentiation. And so they just send out these calls for papers, and people have to contribute papers. They have to come up with a good paper for a certain topic in the area that's relevant to the area, and then people evaluate whether that's worth their time or not. This was from an earlier special issue that didn't happen. It's called Periodicity in the Embryo. And so this was about sort of looking at the emergence of order in space, diffusion in the order of time. So the idea is that there's this tempo and mode of embryonic development, and that, cell division and cell differentiation have these different time scales. So I have a bunch of data from different model systems here that we collected for this, created graphs. So in this case, this is cell births of zebrafish embryo cells during embryogenesis. And then you can see there are these bursts of cell birth, and then eventually the distribution of cell births becomes more diffuse over time. But you can get a sense of maybe what's going on underneath the hood by looking at these patterns. This is from cell tracking data for zebrafish. There's also corresponding data from C. elegans, where you have cell births in development. And so it's a little harder to see, but this is the embryo here, where you have a lot of cell births that are bursty, and they have these sort of bursts of activity over time. And then even in the post-embryonic period, which is where the worm is hatched and is still cells are being born because they have a lot of post-embryonic plasticity in certain parts of the worm, there are these bursts of activity as well. And so if we go to the abstract, we talk about looking at sort of these patterns in time and perhaps in space using two model organisms. And so we had the data from the zebrafish and from C. elegans. And those are both based on cell tracking data from development, where you can, track the birth times of cells and figure out like these patterns of cell division and differentiation. And so, based on these findings, we predict that periodic changes in the frequency of new cells over developmental time represent cell proliferation without functional distinction. Aperiodic changes in cell birth frequency will represent a diversity of specialized sublineages. And then to determine whether aperiodicity represents meaningful information or noise, we can use the information isometry technique. So that's this graph here. So this would be an application of this method. This is something that we've worked on in the group in years past, where we create these, generate these plots of developmental cells from trees and look at their patterns. And I don't know if that's really suitable for this special issue, but we can talk about it in coming weeks or offline. But I just wanted to bring people's attention to it. Something I've been thinking about in terms of different, so we need a new project. But I mean, some goal to shoot for here. So we have this. So that's if you're interested in that, let me know. I think Dick left the meeting, so I think we're pretty close to the end of the meeting. So we stop presenting. So before we go, are there any questions or comments? Okay. I would like to say that that we won't be having a meeting next week. We have I have a couple of things going on during that Monday, so we won't be meeting next week. We will be meeting on Friday if Ujjwal and Mayukh want to drop by and give a little update on their projects. It might be a little too soon, but we can stay in touch on Slack, and that should be good. And then we'll meet the following week. And then if anyone's interested in presenting on anything, let me know. We have a lot of spots open. I know we give like the presentations in the first 30 minutes, or we give an update in the first 30 minutes, but there's always space afterwards. Okay, guys. Well, thank you for attending, and see you everyone in 2 weeks.